Everyone, to introduce the work. Yeah, okay. To introduce my work, I'm doing a PhD with a company. I'm with Professor Telvina, Paul, and TECNIFERTI, and my objective has come from the I was already working with a company. I was working in the lab, so this mix provide me the opportunity to explore more what I've been done till now in a more practical way. The objective is to develop a biostimulant, in this case, a BioNPK A. The main objective is the NPK, but I'm doing. I will skip the introduction. Soils are poor. Climate is changing, and we need another options to sustain our crop production. Cost-effective products are the major parameter to have in account, because if it's not cost-effective, if it's not a well-fit solution to introduce in the traditional methods, people will not use it. So the only way we have to take this technology to be able to introduce rapidly in the agricultural market is to go in the direction of the people that produce the crops. So I was telling, my PhD is subdivided in 6 tasks, 7 tasks if we count with data analysis. The first one is about the choosing the strains. There are a lot of strains. There are a lot of bacteria. It's a problem. It's not a problem. The major problem in this work. There are a lot of bacteria with good capacities. I try to choose the best ones from our collection in the lab. As I was saying. Firstly, I will focus in the nitrogen, phosphate, and potassium strains, but I also screen for other abilities, direct and indirect, to after the main biostimulant being developed and well established, I can be able to introduce one more strain and one more strain to adapt the main composition. The second task involves establish a syntactic community, the strains that are capable of produce a trait that we want could not be a well fit to mix with other bacteria, and if they are not fit to mix with other bacteria in the lab, in the field, it's not going to be great. So we want bacteria that are able to be compatible with other bacteria already present and that we are utilizing. The third task, it's majorly what the objective is to find a matrix that can be able to last at least for 6 months. So the production of these microorganisms are not being doing the day before it's applied in the field. The task 4, it's about the application methods from the lab to the field. About the application methods, if it's seed, if it's soil, it's foliar, the mechanisms to quantify the bacteria that we put on the soil, the impacts that they have on the soil, because until now it's not easy to track the bacteria we put on the soil. We can check with the CSS the genera, but the specific strains that we put there, it's not easily trackable. So we need methods to quantify this without mutate bacteria that are not possible. Task 5 involves systematic, involves to develop mechanisms to systematically be able to produce these microorganisms, changing from bacteria to bacteria, utilizing the same production systems. So the contamination risks are big, and we need a way to at least being able to clear, to have a clean product in the end. So the main objective, as I say, is to produce a biostimulant, bioinoculant, under operational conditions. It's mainly focused on maize. I've been trying to do assays on other species. I screened, as I say, nitrogen, phosphorus, potassium, calcium, magnesium, zinc, EA, in the acid acetic, and alginate. It surged from the 1,000 bacteria that Professor Etelvina told I'm staying with just 21. And after this part, I will just do work with 9, the NPK. The first essay was an essay to check the compatibility between bacteria. Some of bacteria like to walk. It's not normal, but when they are in contact with another strains, even in a volatile environment, they spread rapidly. It could be a good response, it could be a bad response, but when they are in the control plates, they just produce a colony and don't have this mobility. This assay, it's a quantitative assay. I weight the colonies, the colony in this case, in the colonies in these cases, in the area to check if there was a promotion between strains when in contact with other or not. The assay per contact, I want to be able to track in liquid media the variations between the strains when I mix them. But the first way I could be able to do some kind of contact was this. I inoculated the base with one of the 9 bacteria, and I inoculated the other bacteria above that layer. In this first case, with one of them, almost all bacteria have a good growth and could be able to grow, less the one. And in this case, we can see an inhibition on growth of the bacteria. The concentrations of them, the inoculum, are exactly the same. From these assays, I could go for 4 combinations from my 9 bacteria that have been able to give me some work with 27 combinations. And we have, in case of the area, growths about more 15% than the control plates. And here we have bacteria in the weight about 80%, 70%. I chose these 3 because in the assay per contact with the other ones, they are all able to grow. From task 3, I've been trying to develop some formulations with some preservants, but it's not going well. I'm trying to store bacteria at room temperature because it's the most effective way to, it's not the most effective way, but it's the most cost-effective way to a company to store their products after production. So I've been trying to do it on some formulations, some references that say that these work, and they do not work for a long time period. I tested this for 30 days. There was some literature saying that it would take like a year, and the formulations sometimes would be even higher than the initial concentration. So I'm not getting those results. From the 3 mixtures that I showed, we can see these 5 strains in relation to control. I choose to, I have a lot of graphs. I choose to show one of the most relevant in case of fitness of the seedlings. We can see that the mixtures of the bacteria, these are the positive controls, these are the mixtures, and both of them now. The 3 mixtures all promote the growth relatively high against when compared to the control. So from here, my objective is just to choose 3 bacteria to have a base to start working on the formulations. But the most difficult task is to scale up the process. Disinfect, grow bacteria on the same bioreactor, grow strains different on the same bioreactor, disinfect the media, establish growth curves, establish the media, a media that can be cost effective. It's a problem, and I've been trying to do it. We have already one formulation developed, it's in trials. I've been tracking the pH of the growths, the turbidity to have a baseline to the 3 bacteria in the media that you use. So if we need to adjust the media for each one of bacteria, now I'm using the same media for all the 3 bacteria. So I want to optimize that process. I want to optimize I don't know how to say, the big enchimento. There are a problem because it's in an outdoor space and we need the mixture to be clean. This is the first formulation. It's already a registered brand. The composition that I tested initially, it's working well. It's not in the numbers that I want it to be, so it needs a little bit more of adjustment. The pH has been stable for the last days, for the last 60 days, which is a good response in room temperature and in the fridge. We have not in INIAV, I promise. I did an assay with potassium bacteria to try to select one. It searched the opportunity to make the assay, and I did it. It's an experimental station, and I'm trying to choose, linking these results from the field to the results from the lab, to choose one of the 3 bacteria. Another assay that I did with in tomato in Agro Global, an experimental field 2, where we have 2 combinations that were able to promote growth, but we can see that even the other ones have almost all of the conditions have a response to the control. The next key steps, I already have been talking about them, but I want to be able to measure the impact and the quantity of the cells that I put in the soil. I don't know how to do it. I don't know how can I track a thing in a so complex system when I put them in the field, but it's a thing that I've been trying to work on. I want to develop the formulation at room temperature. If it's not possible, I will do the fridge. But now I'm working with the conditions that I have. So, and I want to develop the scale-up process and optimize the production cycle, because right now is almost everything manual, and that is not great. So the final objective is to try to take it from the lab to the field. So, thank you.
Thank you very much. Does anyone have any questions? Thank you. Okay, thank you. Please go, Francia. Thank you for your presentation. What is what you call a potassium bacteria?
It's a stabilization.
Your bacteria,
Yeah. They are rhizospheric. From my master's thesis, I isolated bacteria from roots, rhizosphere, and nodules, and I'm now using that bacteria that's present with the results. Yeah. I'm assuming that in free living bacteria we have the opportunity so they can grow without a host specific plant. Because in the case if they were from nodules, would be difficult to make them grow in the soil. So I'm choosing free living bacteria from the rhizosphere.
For your presentation. What is the formulation? The formulation is a liquid or it is
Yeah.
powder or
It's a liquid. Initially, I want to do lyophilized because it was easier, but it's not cost effective lyophilized bacteria in such quantities. So the idea now is to have a formulation that can preserve cellular stability at room temperature. That's it's not working
because
like
I imagine that
yeah
keep the formulation
yeah
good conditions for life.
yeah. I'm aiming for 3 months now. They want 6, but I'm aiming for 3 as a good timing for now. So it's I'm in the beginning of my work, a year and a half. So beginning.
I have a more basic question. Thank you for your talk, it was very nice. I noticed that in the antagonism tests, you weighted the growth.
Yeah.
I've never seen that. How do you do it and why do you do it?
Because the bacteria can grow in different ways, and the area of the colony is not every. If I work with 350 bacteria in my masters, and I can notice that there are bacteria that the colony grows grows up. Some express, some are really thin. So I did the I don't know how to say, but I scrapped it and I put it in microtubes and I weighed the colonies because it would give me a more precise measure to have the growth of the
Thank you. Very nice.
Any other questions? Thank you very much and good luck for the future. It's very exciting.